Good noon. It's about time we call that one. We're delighted to see all of you here in the room and in the Zoom. Hello. And we're also delighted to have with us today Matheus Januário, Ph.D. candidate from the Roboski lab. We're most well known for his fabulous mustache, but also as a model graduate student. Matheus is the proud recipient of the Laventin Award, as well as having currently a paper invited and a paper in review for Ecology Letters and American Naturalist, respectively. He's here to update us about his dissertation research about bridging micro and macroevolution, insights from vertebrate comparative demography. Please welcome Matheus Januário.
Can you hear me? Well, okay, cool. Thank you very much, you all, for coming. It's so good to present my work here with my friends and colleagues. And I put a very particular title that I will explain soon. And we will go little by little to explain this very large title here. First, I will start talking about the background that makes me ask the questions that I will try to answer in this talk. Then I will briefly approach what it means when we say that we want or we should bridge macro and microevolution. Then I will talk about possible demographic controls of the speciation rates. I will briefly touch on what I mean on the subtitle when I say comparative demography. And then we'll get to the actual book of the talk when I talk about my first two chapters of my Ph.D. dissertation research, when I explore sexual selection and metapopulation persistence as possible controls of the speciation rate. And I finish the talk trying to answer the question that I put in the title. So first, we will discuss some features of biodiversity that call my attention. One of which is that when we look at biodiversity in very large scales, what we found is usually that it's very nested in ways that are unexpected by neutral models. For instance, if you go look in the phylogeny of ray fin fishes, and you look at the first bifurcation, you see that, the oldest bifurcation, and count the species number of those two clades, Cladistia, the shears, and Echinopteri, the other ray fin fishes, what you will count is that they vary by more than 1,000-fold in how many species those two clades contain. If you look inside the Echinopteri lineage, you will find a very similar pattern, which happens again if you look at inside the Neopterigi clade. And I will keep those three clades as a reference here in the next slide, but the point here is showing you that actually this pattern happens in other points of the tree. For instance, when we compare dipnomorphs and tetrapodomorphs and other clades of fishes too. So this is actually a somewhat recurring pattern when we look at large phylogenies. Species are unevenly distributed in clades. Sometimes this varies by 1,000-fold. But this is not the only way in which big phylogenies vary in a way that calls my attention. When we look at speciation rate and its distribution among big phylogenies, and here I'm showing the speciation rate of some lineages. The ones painted in warm colors represent lineages that speciate faster, and those painted in blue represent lineages that speciate slowly. What we see is that, again, we see clades alternating between fast and slow rates, blue and warm colors. And I will refer to this pattern from now on in this talk as the phylogenetic structure of speciation rates. And I'm talking about fishes all this time, but I don't think at all that this is a particular feature that is present only in fishes. When we look at the bird phylogeny, molecular phylogeny, we see similar patterns. And the whole question that I'm trying to asking them to maybe answer here is what is creating this phylogenetic structure? So we actually have a theory that explains biodiversity as a whole. It was made more than 150 years ago. And basically, standard evolutionary theory just says that everything we observe in biodiversity is, in one way or another, created by divergence, time, and eventual extinction, which erodes some of the continuities between the demes and create the apparent discontinuities among taxa. So we have a bridge between micro and macroevolution, and it's very well described, not only in the primary literature by Darwin, but also in the literature that came after. So, oh yeah, and of course, if we want, we can even get more specific and get into more details. For instance, Myers ranked those 5 major contributions that Darwin made to our theory: common ancestry, evolutionary change, gradualism, natural selection, speciation. And if we want to be even more specific, we can increase this list, adding, for instance, the other 3 evolutionary processes, the other 3 fundamental evolutionary processes like mutation, migration, and drift. And if we mix this together, we have biodiversity as we see today. So there is no necessity to bridge micro and macroevolution, right? So what people mean when they write papers exactly about this? This one was written or published last year. So this is a recurring issue here. What people mean when we're talking about this? Of course, micro and macroevolution are part of evolutionary theory, but the way that I interpret those types of literature when I read them is that actually we have mechanism-motivated frameworks to explain biodiversity and pattern-motivated frameworks to explain biodiversity. So we're talking about things like the observation that reproductive isolation or phenotypic change can be quite rapid, or that the orientation of phenotypes and genotypes is somewhat aligned with some other types of tendencies we see in higher taxa, versus explanations that usually are using biogeography, large changes in the fossil record, large phenotypic gaps between higher taxa, the evolution of new phenotypes itself, or variation in diversification rates. This last one is the one that I actually can address in the limited time that I have doing my PhD, of just many years. But still, the point being here is that if we want to bridge, and by that doing a very specific connection between a mechanism-driven a framework and a pattern-driven framework, what we are looking for is some microevolutionary quantity that will allow us to predict the speciation rate, and so maybe explain part of the phylogenetic structure of speciation rates we observe. So one of the most successful frameworks, in my opinion, in trying to help how we study those things in a more direct way was built by Warren Allman in 1992, but you can see pieces of this reasoning in other parts of the literature, at least since Mayer. So Allman conceptualized speciation as a non-instantaneous process, and so we can address the different major factors, or better saying stages that speciation has to pass through to occur, which allow us to study speciation as a dynamical system in which we can dissect the contribution of different components to it. So in particularly, Allman, and better saying the incarnation that I'm actually using here, which is a little more recent, separates the formation of isolates as an important possible control of the speciation rate. The rate of evolution of reproductive isolation is another possible control. The rate of evolution of differentiation between those isolates is also important. And of course, those two factors interact among themselves. They interact with the formation of isolates. But whatever the interaction happens here, whatever of those controls are more important for the actual rates, we know that those isolated forms have also to persist for long enough for them to be recognized as a species by integrative taxonomic practice, and which I'm labeling here as completed speciation. Maybe I could have chosen better words for that, but this is what I mean when I say this. What is the advantage of breaking speciation into these frameworks? Well, we basically categorize possible, plausible rate-limiting steps of the speciation rate, which allows us to study them in the maximum isolation that we can. It allows us to dissect them in relative separation, allow us to incorporate ecology in some of them, which I will try in the next slides, and this naturally fits the statistical frameworks that we already have. So coming back to the first time someone bridged micro and macroevolution, Darwin was always emphasizing ecology as a mediator of the of this transition from things that we would call demes to things that we would call clades. In particular, Darwin was focused on highlighting the importance of competition in doing this, but I will actually focus on different parts of ecological theory, notably macroscopic properties that we can observe and measure in populations that I hope capture a little more directly what the major features of demes or populations. And hopefully we can do this under a comparative framework, and so we can compare demographic properties that we see at ecological timescales, demography, under a comparative framework. This is what I'm calling comparative demography here. And how exactly we do this, right? So first we want to look at macroscopic properties that populations have that related to it, that through ecology creates differences between taxa, right? A good first guess, if you ask me, is to look at sexual selection. Also described by Darwin as the struggle of one sex for accessing the other. And we know that if we know some details of the ecological theory and other details of, for instance, if the water in that lake is clear enough for individuals to see themselves and compete for mates, we can, with some stochasticity, predict which lakes in Africa will foster cichlid radiations. So I think that this seems a good first guess or first variable to look at. And we actually have theory that would connect the intensity of sexual selection to speciation rates, especially because very intense sexual selection would promote a faster evolution of reproductive isolation because, for instance, interact with mate choice and the evolution of mate choice itself. There are many mechanisms that would connect those two things, but usually we can make an overall hypothesis here that like the more intense sexual selection is, the more extravagant or the more divergent traits will be, because we have a higher evolutionary pressure for them to diverge in trait space. Or maybe the speciation rate will be faster because we have more evolutionary pressure separating those demes. When we look at the comparative literature, though, what we see is that there is mixed evidence for correlations between sexual selection and speciation rate. And what I would, and the way I would interpret this is that actually what's going on is that we are accessing sexual selection through proxy traits. And sometimes those proxy traits cannot be the best way for we to measure sexual selection. For instance, common variables explored in the literature are the chromatism in birds, but we know that the chromatism can also evolve as a reflection of natural selection for female crypsids. Other proxy used in the literature is sexual dimorphism, differences in sizes among sexes, which also have possibly many strong components. Much of its evolution is also driven by natural selection, actually. So maybe this noise in the literature is a reflection of our noisy proxies. Can we measure sexual selection a little more directly than just using traits? And I think we can. This is the work that I did together with Renato Macedo Rego, a good friend of mine that is now a postdoc in Brazil. And we just submitted this manuscript recently called Evolutionary Ability of Sexual Selection and its Implications for Speciation and Macroevolution. And what we do there is that we measure and compare indices for the opportunity of sexual selection. Briefly, what those things say is that they quantify the skewness in some measurement of success associated with sexual selection. So for instance, let's imagine a population of a certain species, and we then count how we basically summarize the skewness of the mating distribution with a number here. So we count that male, and we access this through some measurement of success, for instance, mating success. So if this is the situation here, we would count that individual have mated 4 times, this individual have mated 1 time, and those other 3 individuals have not mated at all. We can calculate the variance of this and divide by the squared mean, and we will have a number that will reflect this skewness of this distribution here. We can compare this with another different species in which all individuals mate evenly, and so our index will be 0, reflecting the fact that mating success here is evenly distributed between individuals. And of course, we can imagine that mating success is not everything in sexual selection. Mating success is not everything if it doesn't reflect in fertilization success, which is also not anything if it doesn't reflect into fitness through offspring. But the problem is that if you just measure offspring, you were mixing natural and sexual selection, related, for instance, to the ability of offspring to survive after hatching. So the later you measure success, the more you risk mixing sexual and natural selection. And the earlier you measure, the more you ignore other components of sexual selection that happen after mating, for instance, sperm competition. So if we can measure many different types of success, we are basically covering a spectrum of possible combinations of natural and sexual selection. But whatever way we measure success, we can still compare distantly related clades with this, because we can count individuals and accessory indices to those countings. Things that we can do if we are relying on traits such as antler size, for instance. We need a species to have antlers for we to quantify this. But our indices allow us to quantify sexual selection intensity in many distantly related taxa. One of the advantages of approaching this whole problem through the lens of comparative demography. So what we did here is that we assembled a phylogeny of vertebrate orders, like basically grafting the molecular phylogenies of each of those 5 orders of vertebrates into a backbone that I made based on the fossil record. And we collected data on some indices for the opportunity of sexual selection for many different lineages. We did this very systematically, basically following a meta-analytic procedure. So things are reproducible and traceable, which is good when we want to do this type of investigation. And I don't have time to get into the details here, but I'm not only using variance-based indices, we are also studying Bateman gradients, which are important in sexual selection theory, but I will not really have time to address that. If you were interested, we can talk about this later. We used species-specific speciation rates calculated previously by Cooney and Thomas in 2021. And we just use their data set as proxies for speciation rate. We also collected data on bird dichromatism from the study of Jessica Armenta and collaborators in 2008. And I collected sex-specific size data for all species based on the primary literature. And a quick recap here, our prediction is that the more intense sexual selection is, the faster that clade will speciate, or we will see proxy traits signaling the intensity of sexual selection for that lineage. So let's start with bird dichromatism. In the x-axis, we have one of our indices, and I will show just one index per qualitative result. But trust me, they are all more or less following the same general trend. So each slide reflects like a average result of many, many analyses. Okay, in the x-axis here we have our sexual selection intensity. In the y-axis we have how dichromatic a certain bird species is. And in one panel we have the male intensity of sexual selection and the intensity of sexual selection that is experienced by the females of that species. When we add the points, we see no correlation at all. And we are capturing a large difference in dichromatism here. If we want to give examples that use an arbor, birds that you can see in an arbor, we have the red-winged blackbird in our data set, and we also have the house wren. And as you can see, those two species are very different in terms of how dichromatic they are. I myself never could see if I was looking at a female or a male of a house wren, so I don't know, maybe it's me, but definitely they are less dichromatic than red-winged blackbirds. Okay, what about size dimorphism, right? Again, selection intensity in the x-axis. In the y-axis, we will see weight dimorphism. And this whole gray area here means that the males are the larger sex. And the white area means that females are larger. When we add the points, we again see no correlation between proxy traits and the intensity of sexual selection the species experience, which is interesting, right? This was my first working hypothesis. Maybe the proxies are too noisy for we to correctly assess sexual selection, right? We see no correlation between the proxies and the actual selection intensity. What about speciation? Again, we will see in the x-axis the intensity of sexual selection, and in the y-axis we will see speciation rates. When we add the points, we see no correlation for birds, no correlation for amphibians, and actually no correlation for any other group of vertebrates. So again, no correlation between speciation rate and sexual selection intensity as measured by opportunity indices. But when I was exploring this data, I started to look at other types of patterns too. And one of them is phylogenetic signal, which is basically the autocorrelation that some species have due to their shared ancestry. Blomberg and collaborators have described that phylogenetic signal can vary on the type of trait, and that particularly behavioral traits here are slightly more labile. They have slightly less phylogenetic signal than other types of traits. And indeed, in our phylogeny, we can recover phylogenetic signal. It's this blue bar here reflects the phylogenetic signal that I measured using the body weight data that I collected for the dimorphism analysis. So our phylogeny allows us to measure phylogenetic signal in a way that is aligned with other results in the literature. What will happen if we take the same phylogeny and measure the phylogenetic signal of the intensity of sexual selection? Well, what happened is that the intensity of sexual selection has remarkably small phylogenetic signal, something that deviates a little from the distribution of other traits that Blomberg and collaborators described. So that made me also look at the intraspecific variation in the intensity of sexual selection. Luckily, we have data for many different populations of those species, and we can represent them directly with data points and explore the range of sexual selection that a certain species experience. But to make the data a little less noisy, what I would do instead of showing the points is that I will show the range and an average per species, and I will show the distribution of all vertebrates in our data set as a gray box plot. When we do this, we see that individual species, particular species, with their range overlap greatly with the range of all vertebrates, meaning that it's not only that sexual selection intensity is highly labile phylogenetically, the intraspecific variation of sexual selection is also very high in both sexes. So, what can we conclude here? Our indices do not predict or explain major contrasts in speciation rate, but sexual selection can still be important for the evolution of reproductive isolation. What our analysis means is that sexual selection intensity, at least as measured by opportunity indices, is not the rate-limiting step of the speciation process. Moreover, we found that sexual selection intensity is highly labile and variable not only at the vertebrate level, but also intraspecifically. And of course, those indices have limitations. They can vary by sex. They can vary depending according to some sampling choices. And we try to control at least for the largest of these, discarding some data. Those indices also can vary in their ability to recover the intensity of sexual selection if you vary too much the reproductive system of the species. But still, they can reasonably capture at least very large differences, and our power analysis indicate to us that we can recover a correlation between sexual selection and speciation if it was a very strong correlation with an R-square larger than 0.5, for instance, and even 0.3 in some cases. We know that those proxies are imperfect because we explored them through simulations, like in this paper here. Cool, but still I think there is some truthness in our results, some biological meaningfulness in our results, and that is that maybe just mapping selection to proxies is a very complex task, and we should assume less things in further explorations from now on. The trait that a species has is not a direct assessment of how intense sexual selection is for the populations of that species. Another interesting thing that can reconcile this result with standard population theory and population demography theory is that the intensity of sexual selection can vary according to many other factors than just phylogeny. For instance, the social system that the species has, the genetic architecture that the lineage has, the ecological feeder in which individuals interact, among other factors. Okay, this is interesting to study sexual selection, but I was actually trying to explain or predict speciation rates, which means that I have to come back to my chalkboard and look at other possible rate limiting controls of the speciation rate. Well, Harvey and collaborators have looked at the literature, and what they have found is that persistence is the least explored of all these factors here. So I think this was another way of maybe bringing a new result to the field. So we are looking to answer if persistence is a major control of the speciation rate, a work that I did together with Professor Malin Pinsky from University of California, Santa Cruz. And what we have learned in the first chapter here is that for our estimates to be meaningful, they have to be comparable across species. We have to be able to measure them. They have to put a number into them. And we want them to reflect persistence, of course. And they have to have phylogenetic signal because speciation rates have. And of course, they have to correlate with speciation rates. And what I mean when I talk about persistence, it's really hard to sort clades in which will persist better, right? They're all alive and they're all persisting and they are surviving disturbances in the environment. So how could we study this? And the way that I decided to approach is that I decided to measure other types of macroscopical properties that metapopulation theory would associate with a particular way in which lineages persist. So we are not measuring how much they persist, we are measuring the way in which they persist. And how exactly that translates to a number, I can explain this by if we make the mental experiment of taking a metapopulation that is at a certain equilibrium of patches that are occupied, and we somehow make a disturbance that will extirpate individuals in many of the squares in this lattice, and we will observe what happens after that disturbance, how the metapopulation recovers. Of course, the long-term tendency is that this metapopulation will recover a certain proportion of patches occupied, and one of the features that I am measuring is exactly this proportion of patches. And if your spatial grid is very, very large, this basically is a measurement of how common that species is. And you can also measure how fast this species recovers this equilibrium state, which is somewhat a measure of recoverability. So for instance, in environments in which disturbance is frequent, you would expect that it's more advantageous to be a more recoverable species. This means that we can classify species not only how fast they recover the equilibrium states, but also the equilibrium states themselves. And moreover, we can align those two properties and construct what I'm calling a persistence space, which is basically a bivariate plot. And we can thus basically classify species in their modes of persistence. If they are common and recover fast, if they are rare and recover fast, if they are rare or common, and if they are slow or fast. Cool. Of course, this is a continuous measurement, right? I'm illustrating this with the extremes, but we are measuring this continuously. And what are the advantages of doing this? Well, by measuring those macroscopic quantities, we can compare species more directly because, again, we are not depending on traits. This maps into properties that we associate with persistence, at least in very simple metapopulation models. And they are simple enough that we can estimate the parameters with simple Markov models. And another good feature of this is that we can base our estimations in the direct observation of some extirpation events. Which type of data would allow us to do that, right? Well, we have long-term surveys of populations of demersal fishes from the US continental shelf. We are encapsulating 9 different biogeographic regions, the different colors in this graph here, and each of those regions were sampled, have different numbers of population sampled or places in space that were sampled. They vary. You can clearly see like those regions here have more points than other regions here. But regions don't vary only on that. They vary in how many species we sampled in each one of them. They vary on how frequently those populations were sampled, for how long they were sampled, between 10 and 31 years. So many things vary between our different sampling units here. And we have to account for this when we compare persistence. The original data and most of the estimates that I'm using, including like some data treatments that had to be done before we fit our models, were done by Bat and collaborators in 2017. So the way the data was actually obtained was through bottom trawling, which is basically dragging a very large net in the bottom of the ocean and taking a snapshot of the fish community that was there in that time and year. And we based demersal fishes are fishes that are associated with the bottom. And we based that in cells that had a certain number of minimum toes, which is those dragging sampling points. Cool. So I hope I have convinced you that we have meaningful and comparable measurements of persistence modes. And now the questions that remain is, do these things have phylogenetic signal and do they correlate with speciation rate? Well, if those persistence modes had phylogenetic signal, we would be able to plot them in the phylogeny and see that clades will clusterize in those syndromes here. So we will see a blue clade, a black clade, and a red clade, for instance. And when we do this in the original data, this is somewhat what we see. We see blue clades here, we see red clades here, blue clades here again, blue here, red here. We do see that. And we actually can make formal statistical tests, and we found significant phylogenetic signal in those two properties here. Moreover, another way to explore phylogenetic signal is that we can basically partition the variance in our data, very similar to a ANOVA test, and we can do this with things that we know if we shade sample. I don't have time to explain exactly what those things are, but we do simulations and we show that they are important in our sampling, and they explain some part of the variance in those two components of persistence. We also know that biogeographic regions are associated with a certain percentage of the variation in our data. But, and again, I can't stress this enough, our sampling does vary according to biogeography, so we cannot completely point out that those things are true biologically meaningful signal. It could be sampling still. But we have some variance in our data set that is aligned with the taxonomical rank of that species. So family, genus, species, and sometimes the interaction between the biogeographic region and the family, which is an independent way of evaluating phylogenetic signal and comparing with other factors. Moreover, this is a very complicated graph, so I will just touch briefly on it. But basically, what we can also do is that we can measure in a third way the proportion of variance that is explained only by phylogeny. And depending, those things don't represent necessarily anything. They're just different sets of species right now. Don't care too much about the colors, but like depending on the set of species that we measure, we have different amounts of variation that is explained only by phylogenetic relatedness. What happens if we add the traits? And can we increase our predictability in terms of persistence modes? Can we improve our ability to predict the persistence mode of the species if we have the phylogeny and the traits? In this next plot, I will show the residual variance that is not explained by phylogeny here. And what happens is that usually we can't. We don't learn much if we add the traits once we already have the phylogeny. Okay, but what about speciation rates? This was the whole point. Seeing if those persistence metrics correlate with speciation rates. Even though we cannot predict them with traits, at least we can see if we can predict the speciation rates with them maybe. And we have a lot of phylogenetically independent variation in speciation rates. So again, as we saw in the first pictures that I have showed to you with blue and red clades, now we have purple and orange clades here. And what we want to know is that if particular regions of that persistence space are associated with specific speciation rates. For instance, if you believe that stability is what would allow clades to speciate more, the prediction is that lineages that recover fast and have a larger number of patches occupied at equilibrium will be on average more stable. And so this would be the prediction associated with this particular interpretation of how persistence will translate to speciation rates. But we cannot really figure a very specific hypothesis on how speciation rates should be distributed on this purely by ecological theory, so this is just an exploration. This is one of the possible patterns that we could find. The real thing that is important here is if the test is significant. Not necessarily, remember, this is a first exploration. And what we do here is plot species in this persistent space. Our turnover or recoverability measure is in the x-axis, our commonness is in the y-axis, and then we'll color the points based on their speciation rates. When we do that, we see no significance at all. We cannot predict speciation rate using persistence modes. And we can even highlight this pattern even more if we just plot the 20% slower and then 20% faster speciators in our data set and see that they are scattered through the whole persistence space. Okay, so some conclusions on persistence here. They do not predict speciation rates, but they are weakly correlated with many traits. I didn't approach that in details. We can talk about that later if you want, but I am unsure if the correlations are strong enough for a way to assume things from now on. But they are correlated. This could be an artifact from the phylogenetic signal that we know that persistence syndromes have. What we know is that persistence syndromes have phylogenetic signal, and they vary according to biogeographic region. More general conclusions on comparative demography is that some macroscopic properties that populations have, some of them have phylogenetic signal, for instance, persistence. Some of them don't have, like the intensity of sexual selection. And they raise more questions, like how much of the variation in those properties that populations have are aligned with biogeographic regions, with climes in space or resources, or with abiotic variables. How much of that is correlated with things that we know and things that we will possibly, hopefully, find out later. The interesting things that maybe we can explore that in shallower phylogenetic scales, where we can apply dedicated experimental designs and incorporate the most information that we can about the ecological theory in which those individuals are interacting. What we know for sure is that they don't predict speciation rates, which is aligned with many other results in macroevolution. Like when people measure the evolution of reproductive isolation between populations, they have found that they do not predict speciation rates either, which was later confirmed by a different group of researchers, also in birds. When people measure the rates into which these evolve geographic genetic structure, what they found is that this also does not predict speciation rates, which was also confirmed by different researchers in different studies. So what does that mean? Like, what if we keep finding this, that no demographic property allows us to predict macroevolution? What does that mean? This is a very big if, and I cannot stretch how much I'm speculating here because I have no crystal ball. I don't know. Maybe tomorrow we can figure out a good proxy. And in a certain way, I want to understand macroevolution, so I would love to see one. But what matters if we can do this? Does this mean that micro and macroevolution can be bridged? No, they are bridged since the origin of species, right? What means that we have to look at other types of factors that maybe can allow us to predict diversification rates. So basically, we are refocusing on classical occurrences that macroevolution already has. I'm talking about things like historical contingencies, developmental biases and structure, or the hierarchy itself. Those are things that macroevolutionary theory already studies, as highlighted in this table here that Jablonski made in 2020 and that I painted accordingly. But macroevolutionary theory is more than that. It has many mysteries that we are not really sure how or why they appear. And I hope we figure this someday. But a real possibility is that our data is too sparse or too information poor for we to figure that out. And just to quickly point this out, of course, there are more components of macroevolutionary theory. So what is the importance of biogeographic rounds, of the ability of evolve or to respond to natural selection? What about how frequent and how important are what Jablonski and collaborators call perfect storms, clades that have all the factors that we associate with faster speciation rates? Those things we don't know yet, but we have to figure that out, I hope. Well, but before I end here, I want to thank everyone that helped me do my research, my supervisor Dan, my other colleagues in my laboratory here, my committee members, Nate Sanders, Gideon Bradford, and Johannes Fofopoulos, everyone that helps me in other aspects of my research, everyone that funds me to do the research that I like, and all the thousands of people that collected the primary data that I used in my analysis. Thank you very much. I can take any questions you might have. Nate.
Why does anything have to predict speciation rate? What if it's just haphazard and...
What if what? Sorry?
I'll use the microphone.
Oh yeah, that's true.
Just because I want to throw it next. It's not. So the question was, why does anything have to predict speciation rate?
Well, why it has to predict speciation rate? Because I want to know, and individually because I want to know, and because I think we have to look for theories to explain the patterns that we see in biodiversity. What can we learn if we have a theory of what generates biodiversity? We are so far that I don't know even if it's fair to speculate what we will earn. But the things that we look at biodiversity, we see patterns that are interesting and we want to figure out why. Me individually am driven by that. I just really want to know why there are so many birds of a particular kind, so many fishes of a particular kind. Yeah. Kristen.
Thanks. Really great talk. I have actually a lot of questions, but I'll just skip them.
Yes, I can use the mic. Okay. Okay. I'm curious about, for your first project about the intensity of sexual selection, I'm wondering, I think it's a really interesting way to go about trying to capture sexual selection intensity as the variance in reproductive skew, but I'm wondering if you think that captures something different than maybe how we might just classify like mating schemes, or mating systems rather, the degree of monogamy versus polyandry versus polygyny.
Yeah, this is a very good question. So what we know is that if you change too much, like to very extreme mating systems, you will bias the index value numbers, the values of the indices themselves. So there is some variation that is related to that, and we try to control for that. So we remove the largest factors from our data set. We work counting them more in the more representative universe, if you think in that way, or less noisy universe, better saying. But this does vary, and I am still figuring, because this does reflect real differences in mating systems, right? Some mating systems, like forced monogamy, for instance, or harems, will create different amounts of intensity of sexual selection inherently. This is true. I think our indices capture that partially, and it's really it's a short blanket, right? If you take too much variation in mating system, you know that you are working with indices that might not really rank well, species in terms of the relative intensity of sexual selection. But if when you are restricting your universe, you cannot account for a lot of other variation, right? So we have to figure a balance between this, and we decided to first reach this balance through a more comparable universe first. But definitely there is variation associated with mating systems, and we cannot account completely only for that, only with instances of opportunity for sexual selection. Yeah, that's true. So it's true and it's complex. That's the classic answer in biology. It depends.
That was really fun.
Oh, thank you.
I was wondering if
Thank you. Okay, that was really fun. I was wondering if you've looked at, with your measures of persistence, if you've thought about extinction rates.
Estimating extinction is very hard.
Yeah, so it's just too hard and we can't.
It's very hard, yeah. Like from molecular phylogenies, we know that we cannot trust, right? And using the fossil record, well, it's again noisy because you have whole problems on estimating age, and so you have large confidence intervals on durations, which mean large intervals of extinction rate estimators. And also, there is a blend between what we mean when we say persistence of isolated forms and extinction rates of full-fledged species, things that all taxonomies would recognize as separate species. Are they the exact same phenomena? I'm sure that there is a continuum between them. And what we try to do is that we try to work much more on the continuum, on the part of this continuum that is more persistence than extinction. With that said, in that spatial scale, if you are a very common species, that will be associated with range size, which we know is probably the best predictor of extinction probability. So we expect them to be associated, but again, we try to separate extinction and persistence. And another sad part of this is that we can only fit the Markov model in species that have a lot of data, which usually are not the rarest species that exist. So there is, again, a whole spectrum of things that we cannot capture, not because we are not thinking on this hard, but because we need data to estimate these things, and collecting data is so hard. Yeah, so that's it.
We have some Zoom questions. We have some Zoom questions. I think you inadvertently answered one of them. What is the relationship between persistence and range size for your data set?
Yeah, range size correlates significantly with commonness. But in this scale, they should, right? Like if you were a species that occur in like 3 patches in that scale, you have a small range by definition, right? So there is a kind of like automatic correlation between them. So that's why I don't think necessarily that is super interesting. It's hard also to separate noise from signal in this particular correlation, but we do evaluate, and it is significant.
Fantastic. Also, hey, Matheus, great talk. I love the way you have systematically evaluated all these ideas about connecting organism traits to estimate speciation rates. General question, what do you think is the correct quote-unquote null hypothesis here? That speciation rates are just random, idiosyncratic? In that case, speciation rate may be a product of something abiotic?
Yeah. Okay, dissecting the layers here. I cannot say that they're not random if we have not figured why, right? So I think it's open empirically, but I tend to think, I don't think they are. Like when we look at the fossil record, we see speciation rate varying through time according to some factors, sometimes abiotic, sometimes biotic too. Like for instance, after the angiosperms emerge, you have a cascade of changes in ecosystems, and you can call this biological or abiotic depending on how you frame particularly. But anyways, we know that speciation rates vary. I don't have the generator model to tell you that they're not random, but we have a hard task ahead of us if we want to be really precise of like, okay, if they are not completely random, what is structuring that? That was the question that motivated me, and I don't have the answer yet, unfortunately. So it varies according to some abiotic factors. Even extinction itself. Sometimes they vary together.
Second question, I wonder, with the wide availability of whole genomes, have you considered proxies that may be measurable directly from sex chromosome data? Exempli gratia, the relative effective population sizes of sex chromosomes should contain some information about productive skew. Would this be a more direct than the proxy based on mating success?
That's a very good working hypothesis, if you ask me. We have genomic data, but I don't know if someone simulated to be really sure if the genomic measurement would be a good proxy. I think this is the first question that we have to answer. I tend to think that this is plausible. I talked with some people that decided to measure sexual selection intensity in that way, and they have been telling me that they were being reasonably successful in terms of their expectations of how the data should look like. I think a formal analysis does not exist yet, and if it exists, I don't know. I haven't read. So definitely is another way that you can compare many species within the same type of measurement. So that's a possible way forward. That's correct. But I don't know exactly how. Ethan?
Just very quickly. I can catch just fine. I'm wondering if it looks like most of what you're doing right now is just looking at relationships between two variables. And I'm wondering if there's, obviously these things are interacting with one another. Have you tried any sort of structural equation modeling or sort of multivariate approaches to seeing how different variables are collectively affecting or modulating one another in that sort of scheme?
This is a possibility. I tried to do that for the persistence prediction. So I used sets of traits to try to predict persistence. And I found that morphology was the only one that was significantly predicting in a multivariate, like using multiple variates to predict persistence. But when I opened the hood of that result and trying to figure out what was going on, it was weird, right? The morphology, especially the coarse morphology data that we have in FishBase, was predicting persistence. And what I think I found is that it's the phylogenetic signal of morphology that was creating this artificial correlation. So doing multivariate analysis is not that straightforward because we know that a lot of things will covary simply due to relationships of kinship, or like phylogenetic related. This is what I want to say. So it's not straightforward, but from a biological theory standpoint, yeah, we should try to think on ways to properly evaluate this. This is a good possibility. I was going little by little because I need time, to believe in the things that I'm measuring.
I was expecting a complete multivariate analysis today. I'm very disappointed. Thanks, I appreciate it.
Okay. Any questions? Oh, here now.
That was great, Matheus. I love the ambition of what you're trying to do. You caught my attention with what seems to be a total dismissal of morphology as a correlate to perception as phylogenetic signal, especially coming from fishes. Mm-hmm. One thing you haven't emphasized much in what you presented is natural selection, and there's a huge correlation between function and ecology in fish morphology. And I realize how complicated it is to do multivariate analyses at that scale, but I think that's something worth exploring. Certainly not saying, oh, well, it's a bunch of phylogenetic signal and that's that. Mm-hmm. Especially because you're looking at a huge phylogenetic scale, and there's a huge amount of variance in morphology across vertebrates and across fishes. A lot of it is going to be convergent, but most of it is adaptive. Mm-hmm. And so I think persistence being correlated to morphology is not something you should simply just say, it's phylogenetic signal.
I would agree with you, but I don't trust fish-based data that much. That's more an affirmation on the nature of the data. If I trust more on the precision of the morphological measurement, and especially if I believe we are using good proxies for how fish interact with their environments.
But I think you should look carefully at what's available on FishBase, because some is more reliable than others. Things like body size are mostly okay, but there are other things that are correlated to life history, others to habitat use and things like that. And it would be worth looking at it a little more carefully, I think.
So like I did a PCA of the morphology, and I saw some points clustering, and they were clustering in terms of their persistence syndromes too. Amazing, right? Morphology. And then I made the points look like the family, and what I found is that the clusters I was seeing were just families, and we know that persistence has phylogenetic signal. So that's why I suspect this might be an artifact yet. But definitely I think I should look closer. It would be amazing, right, if we could take independent measurements and test this, because we would be able to like independently measure the things that we want to study. That would be amazing, and definitely we should explore this. I was just stuck with the fish based data that I had at the moment. But it would be really cool if we find that this is robust. I was not dismissing morphology.
Just to follow up on that, it's interesting to think about persistence having a phylogenetic signal because it's not something that can be like directly inherited. So maybe this is going off on too far of a tangent, but why do you think it does have a phylogenetic signal? And maybe it's related to the morphology, the family-specific morphology, if that is an important proxy.
Yeah, with the risk of looking too tautological, I would say that like clades are not clades, right? They are not, I mean, what makes them a clade is the set of things that they evolved, and maybe it's a combination of the traits that make them persist in a certain way. What I haven't said is that I made those multivariate species with other sets of traits. So like life history traits, for instance, that inspired the first works in metapopulation theory about like how fast the mode of life history is. And I have found no relationship there. And in other sets of traits, like physiology traits related to metabolic rates, which would be related to how frequently individuals have to expose themselves to acquire more resources. And the only multivariate set that I found was morphology. And that is why I got a little suspicious. It's not because morphology was the one, it was because only morphology was the one. And also, like, we have problems of multiple comparisons too, which I have not really addressed yet. So we have to be careful with that too. But we for instance, like, classify the fish in like life history modes that we know in fish biology are important to classify species, and they are not related to persistence, which was very surprising to me. And as you know, it's Windmiller and Rose's life history modes. Windmiller's.
Was that?
Yeah, Windmiller and Rose's life history modes, we calculated those and they do correlate.
Cool. I'm not surprised.
Yeah. Okay, I think our time is over. Thank you guys very much.